Hi everyone! Today's video is about the iron-responsive element iron regulatory protein system. In short, it's also called the IRE-IRP system.
If you want to skip to any particular section of this video, you can do so by clicking the timestamps found in the description section below. Okay, let's get to the video.
So the iron-responsive element iron regulatory protein system is one of the body's most important cell-level control mechanisms available for iron control. While hormones like hepcidin regulate how iron moves between organs, the IRE-IRP system works inside individual cells. This makes sure that they have enough iron for vital processes like energy production, DNA synthesis, and hemoglobin formation without allowing dangerous amounts to build up within the cell. This system acts after genes have already been transcribed into messenger mRNA. This means that it does not decide whether the gene is turned off or on. Instead, it controls how much of a protein is actually produced from the gene. This gives the cell a fast and flexible way to adjust iron use depending on current conditions. So what exactly are IREs and IRPs? Let's start with IREs, which stand for iron-responsive elements. Iron-responsive elements are small, folded sections of mRNA that form a stem loop shape. These structures are found only on mRNAs that code for proteins involved in iron handling. Examples include mRNAs for transferrin receptor 1, which brings iron into the cell. It also includes mRNAs for ferritin, which safely stores iron. It also includes mRNAs for ferroportin, which exports iron out of the cell. These IREs, which are sections of mRNAs, can also be found on mRNAs for several enzymes required for heme production as well. Now let's talk about IRPs. IRPs stand for iron regulatory proteins. There are two types, IRP1 and IRP2. They're proteins, so they float freely inside the cytoplasm. They attach to IREs, which are small folded sections in the mRNA. Whether the IRP binds or not depends on how much iron is present inside the cell. This way, the IRE-IRP system acts like an internal iron sensor in a way. It constantly monitors the iron levels inside the cell and adjust the protein production to match the cell's needs in order to bring more iron in, to store more iron, or send more iron out. So let's consider how these would behave in a low iron environment and a high iron environment. Let's talk about when iron's low. When iron levels inside a cell are low, the IRPs, which are the proteins, become active and they attach tightly to the IREs, which are the folded sections of the mRNA. And remember, the IREs are only available in mRNAs that produce iron-related proteins or heme-related proteins. What happens next depends on where the IRE is located on the mRNA. Some IREs are positioned near the beginning of the mRNA in a region called the 5' untranslated region. This comes just before the part that is read in order to make the protein in itself. So what kinds of proteins would have an IRE right at the beginning of the mRNA? The answer is ferritin and ferroportin mRNAs. When IRPs bind in this location, they physically block the ribosome, that is the cell's protein-making machinery. As a result, as you would have guessed, the cell produces less ferritin and ferroportin. This means that the cell is not able or less likely to store iron within the cell and export less iron out of the cell because ferritin is the protein that stores iron inside the cell and ferroportin is the protein that's required to excrete iron out of the cell into circulation. Because this happens when iron levels inside the cell is low, This helps the cell conserve the limited iron it has available in forms that it can actually use, which is why the ferritin, which stores iron, is not produced as much, and ferroportin, which exports iron, is not produced as much as well. There are other IREs as well. Um, Other IREs are located near the end of the mRNA, that is the 3' untranslated region. These IREs are found on mRNAs that create transferrin receptor 1. In this case, when the IRPs, again, remember IRPs are the proteins that are floating in the cytoplasm, when IRPs bind to IREs that are at the end of the mRNA, it does not really block the protein production. Instead, it protects the mRNA from being broken down by the cell. And therefore, the mRNA lasts for a longer period of time. Because the mRNA lasts for a longer period of time, it can be used repeatedly to make more proteins. And this leads to having higher levels of transferrin receptors on the cell. With more transferrin receptors available on the cell is able to pull in more iron from the bloodstream. Therefore, in a low iron state, you're bringing in more iron. So, together, the two types of responses that I mentioned that reduce iron storage and export while increasing iron uptake from circulation allows the cell to maximize iron acquisition and minimize iron loss when iron is scarce in the cell. Now, let's talk about what happens when iron levels inside the cell are high. When iron levels inside the cells become high, the system switches directions to prevent damage from excess iron. So in this state, IRP1, which is again the proteins that are circulating within the cytoplasm of the cell, changes its shape. The way it changes shape is by binding to an iron sulfur cluster. This binding and changing of the shape makes it unable to attach to the IREs anymore. And remember, IREs are specific sections of mRNAs that specifically create proteins that are needed for iron homeostasis within the cell. While this is happening, remember, I also mentioned that there's something called IRP2, which is again another protein that is floating within the cytoplasm of the cell. And IRP2 is marked for destruction and broken down by the cell, with fewer IRP2s and IRP1s being inactivated. The mRNAs for ferritin and ferroportin are no longer blocked and can now be translated normally. This results in an increased production of ferritin, allowing the cell to safely store extra iron, and increased ferroportin, which helps move some of the iron out of the cell. Meanwhile, transferrin receptor mRNA is no longer protected and becomes unstable, so it quickly disintegrates inside the cell. This reduces the amount of transferrin receptors made, and therefore, the cell is unable to bring in extra iron into the cell. These coordinated changes prevent additional iron from entering the cell while encouraging storage and removal of excess iron. All of this protects the cell from iron overload. The IRE-IRP system does not respond only to iron levels. It also integrates other signals such as oxygen availability, oxidative stress, and the cell's energy state. For example, low oxygen levels can influence IRP activity in ways that support increased iron use during red blood cell production. This ability to respond to multiple environmental cues allows the system to fine-tune iron metabolism differently in various tissues and under changing physiological needs. Disruption of the IRE-IRP system can have serious consequences for health. If regulation fails, cells may not receive enough iron to function properly, contributing to anemia, or they may accumulate excess iron, which ends up damaging tissues and is linked to neurodegenerative diseases and other mitochondrial dysfunction. Because iron is essential yet potentially toxic, even small defects in the intracellular regulatory system can interfere with cell survival and organ function. So, what's important to know about the IRE-IRP system? The IRE-IRP system is a sophisticated intracellular feedback mechanism that carefully adjusts iron uptake, storage, and export according to the cell's immediate needs. By controlling the stability and translation of mRNAs that encode iron-handling proteins, it ensures that each cell maintains a safe and functional iron balance. It works alongside large-scale regulators such as Hepcidin and transferrin. The IRE-IRP network forms a crucial layer of protection that supports normal metabolism, oxygen transport, and overall health. And that's it for this video. I hope it was helpful for you. If it was, please make sure to subscribe and like this video. If you want, please go back to my channel and check the other videos about other iron-related proteins that will be helpful for you in order to understand this video better. Thanks so much. Okay, see you in the next video. Bye.